Remember we talked about the fact that the human genome is constant. Remember we talked about the fact that the only thing that we know moves in the human genome are these transposons, and they move over evolutionary time. Do you remember that, Tom? Remember I said, but there's one case in which you have an arrangement of genes in your blood cell which is different than what is in your sperm and which is in your female counterpart's eggs. And those deal with antibody genes. The structure of your antibody genes in your blood cells is different than the structure of your antibody genes in your germline. And that's because after conception, after the formation of a zygote, during embryogenesis and fetal development, in these B cells, the antibody genes are rearranged in the chromosome. And each B cell forms a specific antibody because it has a specific reorganization of a heavy chain and a light chain. And it occurs by shuffling groups of genes, that is actually shuffling them, cutting the DNA and rejoining them in different combinations during the development of the blood cells, such that if you looked at the heavy chain and light chain genes in each of these 4 cells, you would find that they would be different in organization slightly and different in sequence. Because the only way that you can make a different antibody is to have antibody variable regions, that is, amino acid sequences that are different. Eden, did you understand that? I'm going to go over it a little bit more. Do you understand the concept? The point here is, how do you get a billion different proteins from 2 different genes? Think about that. The only way this can be done, and Stephen's question was very incisive, is to start rearranging genes, meaning mixing and matching and changing their organization in the chromosomes in different cells during the development of those cells. And there's very specific, I want you to hold your question, Julie, because I want to show you how this happens, and then I'll throw it open to questions. There's very specific enzymes that do this, and there's very specific sequences that do this, because everything works by sequences, and everything works by enzymes, nothing works by chance, and here's how it works. Here is, let's just say, a heavy chain, something that'll give rise to a heavy chain. And there's a number of different genes, we'll call them genes, that are involved. And they're all next to each other on the chromosome. Notice that we have 100 different, what we would call variable region genes, 100 different variable region genes or sequences. You can call them whatever you want. We have 30 different, we call D segments, and we have 6 different J segments. 100 different V segments, 30 different D segments, and 6 different J segments. And each of those has a different sequence. And each of those would code for a different amino acid sequence. And furthermore, then we have slightly different coding regions of what we would call the constant region. We have this, and this requires 8 genes. Conceptually, you could look at these DNA sequences as saying, these are the DNA sequences that make the variable region. These are the DNA sequences that make the constant region. And during development of the blood cells, what happens is that one of these joins with one of those, joins with 1 or 2 of those, and joins with one of those, and they all come together in the chromosome. This is not a splicing. This is not splicing. This is simply rearranging in the chromosome these particular sequences. And so then in one beta cell DNA, this is what's in the germline. We have a rearrangement of the DNA, and now in one beta cell that's being formed in the embryo, We have this specific sequence, that specific sequence, that specific sequence, and that specific sequence. That's then transcribed, and then splicing can occur to get rid of one of these, and we have this sequence, that sequence, that sequence, and that sequence that makes a specific antibody. And furthermore, if you take all the possibilities here, so 100 of these sequences, 30 of those sequences, 8 of those sequences, 6 of those sequences, 8 of those, if you put together those combinations, it's 144,000 combinations, just to make the H chain. The light chain has a similar organization, and you can have 144,000 combinations of L chain. Since you need an L chain and an H chain to make a specific antibody, use 21 billion different combinations. And those 21 billion different combinations, Stephen, all occur. Every single one of them happens by chance arrangements in the genome by very specific enzymes that do this. One B cell, one H chain, one L chain of one specific kind makes one antibody. And in your blood system, when you're born, you then have a unique H and L gene in every single solitary different B cell. Is that clear, Max? In your germline, in your sperm or your egg, you have the original gene. You have to, because it's passed on from generation to generation. So this is called a somatic cell DNA rearrangement. If you did a DNA blot, Elaine, and you compared the structure of the antibody gene in, let's say, the germline DNA, and you compared that to the structure of the antibody gene in one of your blood cells, you'd see a difference. You'd see immediately a difference because they're different, because there's a rearrangement. And that's how that was discovered. In fact, some Nobel Prizes were won for discovering this mechanism because people asked the same question that you did, Stephen. How does this happen? It was always a big mystery. So this is the only known case in which specific DNA sequences in the human genome in your somatic cells have a different organization and structure than in your germline, because they're passed on in the germline, and what happens in the somatic cells has to be able to make these different antibodies. Julie, what's your question?
So does that mean that...
I can't hear a word you're saying. Thank you.
Does that mean that the DNA in the A part where it says DNA rearrangement, those are the cells that are in, that's the DNA that's in every cell but your blood cells?
So this would be the DNA that would be in every one of your cells
but your blood cells.
but your blood cells.
That's what you mean by
and
germline.
not in all your blood cells, just in these beta cells, the ones that give rise to antibodies. Your red blood cells don't produce antibodies. Okay? So there's very specific cells in the blood that gives rise to the antibodies, and this rearrangement occurs in those kinds of cells. So like the B cells and these T cells, but not in your red blood cells. So that's correct. So in your skin cells, you have this rearrangement. This is a very cell-specific phenomenon.
So does that mean there's one of the sequences that tells how the DNA or how the embryo or how development's supposed to go during...
You have to ask a more precise question.
Is there a sequence that tells the DNA to form like that in just those
Absolutely.
cells? Okay.
Absolutely. So imagine that this only occurs in these beta cells, B cells. So the enzymes that do this are only present in those cells. And so the genes that encode those enzymes have to have particular switches that are only turned on in those cells and not turned on in the skin cells.
But is that constantly happening, or does that only happen during development?
Only happens during development. In other words, after you form your blood system, it's formed, so to speak. Okay? And so when an antibody comes along, lo and behold, in your population of these B cells, the antibody comes along for a particular, let's say, for smallpox virus, let's say that's the antibody that recognizes it, it's going to then all of a sudden say, ah, I see the smallpox virus. And now it starts to divide. We'll use the red one. Now it starts to divide, and you make tons of them. And you make lots of them. And it then goes and tries to clear the smallpox virus and gets rid of it. Sometimes the virus overwhelms it, and you just can't counteract it with your immune response, because we do get sick. Have you ever had the flu? You have antibodies to flu viruses, but you get the flu. It's the virus overwhelms your system, but eventually the system takes over and you get better, right? So eventually you start producing more and more antibodies in order to get better. But that's essentially how the system works. Stephen, in a nutshell.
Are those the combinations static in a person's blood cells throughout their life, or does the immune system learn new specific combinations or new
good question.
specific antibodies that aren't in the 21 billion?
The 22 billion is all you need. So this is a random phenomenon. So the potential, the cells that produce let's say the cells that produce all of those 22 billion antibodies are sitting there and waiting for something to happen. You may only have a few of them, but you have them. When you are, let's say, assaulted with a particular antigen, it will recognize that that antigen is present, and the cell producing the antibody to that antigen might be present only in a few hundred types. Then it starts to divide over and over and over again and fights that antigen off. Okay, wait a sec. But now, let me finish. But now what happens is that since you've seen that antigen, the cell that produced that antibody is now not present in just, let's say, a few hundred cells in your blood system. It now be present in millions and millions of cells because it has a memory. And so if you're then assaulted with that same antigen again, you're going to be able to clear it much more rapidly because it's seen it before.
That wasn't my question. I didn't phrase it well.
Okay, go ahead.
What if there's a pathogen that is not recognized by any one of those 21
Doesn't
billion?
happen. It's just not going to happen. I mean, there's just so many different combinations. There's only so many pathogens that are around. This is a random thing. And so in general, I won't say that it won't happen, but in general probably doesn't. You're going to be able to elicit a response, but again, you might not be able to elicit a strong enough response in order to overcome what the pathogen is doing, because people do get sick and they do die, okay? And that's where vaccines come in, so you can boost this whole thing and prevent the infection to begin with.
Okay.
Eden. I can't hear you.
With the antibody plate test that
With the antibody what?
The antibody plate test that scientists do to detect if a desired clone is on a plate.
Yep.
So they use
Do you mean,
insulin.
is it in the context of an infection or a clone or what?
No, sorry. It's in the context of doing recombinant DNA.
Yeah.
And then finding the desired clone.
Right.
They use an antibody test.
Yeah.
So does that mean that they can't make an antibody? If they want to find a certain protein, can they make an antibody?
Who's they?
Scientists, sorry. So do we have an antibody to insulin?
No, the antibodies that are used are generally made in rabbits.
Oh, okay.
Okay, they're not made from human beings, they're made in rabbits. You take the protein, like if you wanted an antibody against protein, against insulin, you'd inject it into a rabbit. The rabbit would see it as a foreign protein and would elicit a response, and then you'd purify the antibodies from the blood serum of the rabbit.
Okay.
That's how it's done.
Okay.
It's done experimentally. Dora, do you have any questions? Faria, do you have any questions?
No.
It's a fascinating field, this immunology. The point here is that one has to be able to make vaccines in order to counter disease. The point here is that if one understands the pathogens, if one understands the proteins that produce the antigens that infect us, like the coat of a virus, or a certain protein, a toxin protein that's made from, let's say, a foodborne bacteria that causes horrible food disease, one can then counteract these illnesses. And notice this was taken from your textbook, that we have the mortality rate that has gone down significantly because of the advent of antibiotics and because of the advent of vaccinations. And in fact, as you know, you can't send your kids to school unless they're vaccinated against measles and diphtheria and typhus and whooping coughs and brucella, and all of you have been immunized against these things because they are lethal diseases and they're serious. I think it's great that you don't know what polio is. I really do. It's wonderful. But you also should know that there's a whole bunch of anti-science going on there, that people seem to think that vaccines are causing all kinds of problems and have stopped vaccinating their children, which horrifies me and horrifies most pediatricians because one can have a child that can die very easily from just scratching themselves with a rusty nail. And we don't worry about that because we have vaccinations. The point here is that we do need vaccines to many, many different things that we don't have vaccines for now. We'd like to have vaccines, for example, for every virus that one can imagine that would be involved in biological warfare, for example, like Ebola. Do you know what Ebola is, Kathy? Anna, do you know what Ebola is? Hemorrhagic fever. You know what the mortality rate is for Ebola? 99.9%. You know how you die? You bleed to death. Nothing can stop it. There have been some remarkable and tragic outbursts of Ebola around the world. No one even knows what the vector is that carries Ebola, but it generally will wipe out an entire population. And so there's a lot of diseases of which we don't have vaccines for and which we'd like to have them. And there's a variety of ways in which genetic engineering can be used to be able to make a vaccine. The first thing is just genomics, sequencing the pathogen's genome. If you sequence the pathogen's genome, you might be able to identify genes that the pathogen uses to infect its host. Does that make sense? If you can understand what those genes and proteins are, you can counteract them by making a vaccine against those particular proteins. Remember, just like the bacteria virus has to get into the E. coli, and just like the E. coli fights it with restriction enzymes, when we're talking about pathogens that infect animals such as us, there's an immune system that has to respond to it. In many cases, the pathogen tries to overcome our immune system, like the AIDS virus, which I'll get to in a second. But the point here is, if you can understand the structure of the pathogen and the genes that are involved in pathogenicity, you can start to think about targets for vaccines. And so one then can make, using genomics, we can try and identify the targets. How do we identify the targets? Well, here's E. coli H0157. You've all heard of E. coli H0157 because this is what causes major food illnesses, and it's lethal. And in fact, as you can see, this is a very, 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 very serious pathogen. And in fact, many, many, many millions of people get foodborne diseases. And as you can see, 5,000 deaths due to a contaminated hamburger, a contaminated peanut, which is going around these days, peanut butter, contaminated chicken, all kinds of stuff. So cook your meat. This is serious stuff. But this E. coli, H0157H7, a particular strain, is a lethal strain of E. coli. Most E. coli, it's in your gut. It doesn't bother you. So the question is, how do you find the genes that are involved in pathogenicity? Well, you sequence the genome of the pathogen E. coli, and you compare it to the normal laboratory strain of E. coli. And now we're talking about comparative genomics. Clone the genome, sequence the genome, use bioinformatics, compare the sequences, and lo and behold, look at the red. Those are the differences between the pathogen E. coli and the laboratory E. coli, and that's where the potential pathogenicity genes and proteins are. And then by studying what those proteins do in the pathogen, where the proteins are located in the bacteria, and understanding them, one can then perhaps make a vaccine against this particular foodborne bacteria, which is a very, very, very serious disease. Now, in the same context, what if you have a foodborne disease that you don't know what it is? How do you identify it? Well, you identify it in a couple ways. If you know it's some E. coli, but you don't know what its strain is, you then use DNA fingerprinting and compare it to a collection of pathogenic and normal E. coli or Salmonella or Shigella or whatever it is, and you'll be able to identify it by using a DNA fingerprint. This is how the anthrax bacteria was identified that was used to kill a number of people after 9/11. And this is how the individual who just committed suicide, you may have read about this in the news a few months ago, who was accused of perpetuating this crime and who worked in the chemical and biological warfare laboratory for the United States and who was involved in making the anthrax strain that was used to murder a number of individuals. This is how they got him by DNA fingerprinting because they traced the anthrax back to the source and they knew exactly what was the one that was individuals that died, what was the one that was in the laboratories, and lo and behold, we're able to identify presumably the person that committed the crime. But the point here is that you can use DNA fingerprinting to identify these pathogens, and then you can study the pathogen, sequence the genome, and come up with a vaccine. On the other hand, what if you don't know anything about it? So you can't do a DNA fingerprint because you don't know what the pathogen is. Well, nowadays we do this thing using genetic engineering called metagenomics, metagenomics, something that you haven't heard about. But it's very powerful, Derek. What you would do is you would take this contaminated hamburger, you would isolate, just rinse it in water. Isolate every organism on the face of this hamburger. Don't even know what they are. You'd isolate the DNA from this whole collection of bacteria and whatever, and just sequence them all together, collectively. You haven't separated out the organisms. You don't know anything about them. You just put them all together and you've sequenced them, called metagenomics, sequencing many, many, many genomes at the same time. And then you use bioinformatics, these big databases that we have, because so many genomes have been sequenced, you use bioinformatics to sort out what's there, and you try and identify some pathogen that hadn't been recognized before. And that's done in this way simply by isolating the DNA, cloning it in some little vector, and sequencing everything together. And Anna, because the databases are so big, you now look at the DNA sequences, you compare them to the protein sequences, and you say, on that hamburger, I found this, 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 and oh, I found Shigella type 4783. That's the one that does the job because it has a toxin that can cause serious food-borne illness in humans, and now we know what it is, and you've identified it, or you've identified something new. So it's called metagenomics. So the point here is that we have an immune system. The point here is that we'd like to boost our immune system by giving ourselves a vaccine. When we give ourselves a vaccine, the vaccine is stimulating the B cells that are specific for the antibody, for the protein that's in the vaccine that we want to protect ourselves against. If we know what the virus or the bacteria is, we can perhaps make a vaccine pretty readily. If we don't, we have to find out what the pathogen is, and then we have to be able to find out how it causes serious disease. Does anyone remember SARS SARS, that went around and killed everyone? Well, within a matter of weeks, they found out what the pathogen was. It happens to be a virus. They sequenced the genome and found which specific genes were responsible for the pathogenicity, and then made a vaccine in a matter of a year or so to that particular virus, which is pretty remarkable. And so the point here is that you can use these genomics techniques of a variety of kinds to find out not only what the gene and the protein is that's responsible for the pathogenicity, but you can find out a lot of things about how these things infect us. Is that clear, Thomas, conceptually? You're sure? Hannah, is that clear? Okay. Then you can use genetic engineering to make the vaccine, because you have to be able to make a vaccine that can vaccinate us. There's a variety of ways in which that can be done. One of the ways in which it can be done is make the protein in large amounts in E. coli and take the pure protein and then inoculate us, and it elicits an antibody-antigen reaction. A more common way to do it is to engineer the protein that we want to immunize ourselves against in a virus, a harmless virus. And then we infect ourselves, or a doctor infects you in their office with this recombinant virus that has the particular gene that encodes the antibody that you want to be able to be protected against. And then there's a variety of different ways in which this can be done. Bacteria, yeast, viruses, but by a knowledge of what the antigen is, one can be able to make a vaccine. And so here are all the different pathogenic agents for one would like to either make vaccines or we have vaccines, and the one that's proven to be most elusive is the AIDS virus. And in fact, to date, every AIDS vaccine that's ever been made, clinical trials have failed. And it's failed for a variety of reasons. One, because this virus is very smart. How does this virus work? It's a Trojan horse. It gets into these T cells, which are the cells, the other part of the immune system I haven't talked about, and it destroys them. So then you have no ability to fight off this infection, and it destroys your immune system, and you eventually die from other adventitious infections. It's a very tragic and very, very, very horrible disease. It's now controllable with drugs, but still one would like to have a vaccine. Now, one of the reasons why the vaccines don't work is because we vaccinate and make these vaccines so that the B cells in your blood are reacting against the virus. But the problem is that that's not where the virus gets into your system. The virus gets into the system at the point of origin, either in vagina, rectum, or a variety of other places. And that's where the response system has to be attacked. And so now the idea is, instead of making a normal garden variety vaccine to the AIDS virus, like it would be smallpox, that affects your B cells, the idea is to try and make a vaccine that actually elicits cells at the point of entry of the virus, because the thought is that that might be the best way in which one can protect, and or with these things called miticides or microcides, which essentially will kill the virus. I only know this because one of my closest friends, Peter Anton, is head of the AIDS Research Center here at UCLA, and he's the foremost authority on making AIDS viruses and vaccines to AIDS viruses in the world, and he does it by trying to understand what's happening at the site of entry. But in reality, most of the AIDS vaccine trials have failed for a variety of different reasons, mainly because of the virus and mainly because of how it infects. Just hasn't proven to be very easy. Okay. Rose, have I been clear? Samantha? So we have genetic engineering to make drugs, genetic engineering to make vaccines in a variety of different uses. But part of making a vaccine is not simply to counteract a foreign protein, but part of making a vaccine has to do with counteracting a very lethal disease called cancer. And probably one of the most promising treatments for cancer today is using vaccines. And some of you may have read in a newspaper about Senator Kennedy, who has glioblastoma, uncurable brain cancer. It's 97% lethal. Tragically, he will succumb to that cancer because there's very few treatments. But one of the ways in which he's being treated is by using a vaccine that was made against his own cancer cells. And the reason why one can do this is that the cancer cells on the outside of their, let's say on their membranes, have proteins and markers that are different than normal cells. And so if one takes cancer cells and let's say injects them into a rabbit or some other system, one can elicit antibodies to the cancer proteins. And those antibodies, if used in the right way, can be used as magic bullets in order to go to the cancer cell and destroy it specifically without affecting the normal cells. And so one would then like to make vaccines using genetic engineering to cancer-specific antigens, to be able to use them as magic bullets to be able to treat cancer and rid an individual of cancer, which, as all of you know, because all of you know someone who has had cancer, it's a very tragic disease. Both of my parents died from cancer. It was a very horrible thing to watch. And the point is that we would like to get rid of this particular disease by using good science. I should say in passing that because of genetic engineering, we understand what cancer genes are. We understand what the proteins are, and I'll talk a little bit about that in a second. Here's a cell. It divides, as you can see. This cell division, Thomas, would you imagine is highly controlled?
No.
You think it just goes on all the time?
Well, isn't that what cancer does? Just
No,
repeat
but
itself?
in you, in normal.
Oh, it's very controlled.
Yeah, it's very controlled. It's controlled by this thing called the cell cycle. And there it is. The cell cycle has a period of DNA of cell division called mitosis, which you heard about in 3rd grade. And that's how the cells divide. It has a period of DNA replication. Remember, 1 chromatid to 2 chromatids. That happens in this synthetic phase when the DNA in all of your genes is replicated, and has a couple of resting phases. But this cycle is highly controlled. Your brain cells never divide, Marvin. When they start dividing, God forbid you have big problems. Your liver cells don't divide. If they start dividing, you got big problems. Your pancreas cells don't divide after they're made. If they start to divide, you have big problems. So that means there's control points. There's specific proteins and specific genes that control these proteins, and here some of them are, that say when this division can occur and when it's not supposed to occur. And it's highly regulated. And when it goes haywire, you get cancer. And it goes haywire because the genes that control this process become mutated and work when they're not supposed to work, or don't work when they're supposed to work. And so therefore, cells that are not dividing start to divide because this process has been rendered defective. And it's been rendered defective because of the following reasons. And I'm not going to go into this in any conceptual sense, but there's a method to my madness here, and that is these are all of the different processes, in cells that control this division cycle. And that division cycle, Faria, is controlled at a variety of different levels. And I'll just take one for example. You have these things called growth regulators. These growth regulators stimulate cell division. Okay? So if you have to divide, sometimes they will tell the cell to divide. And these growth regulators, again, respond, let's say to a growth hormone or something, by a receptor on the surface of a particular cell type. Again, we're back to signaling. And when that, let's say, signaling molecule, like a growth hormone, binds to that receptor, it tells the cell to start to divide. Imagine that this receptor the gene encoding that receptor mutates in some way, so that that receptor now has a different amino acid sequence. And imagine that that receptor has to change shape when this signaling molecule binds to it. It's the only way it can work. Signaling molecule binds to it, it changes the shape of the protein, and a signal is sent, divide. But imagine that the gene in that cell has mutated. And let's say it mutates so the amino acid is changed. So now the shape is changed, even though that signaling molecule is not there, and it starts sending a signal to the cell and thinks it's dividing. It's supposed to divide, and it divides. And that's how cancer starts. It's one way. One way. But there's many other ways that I'm not going to get into, conceptually. Imagine, though, if one could be able to block any number of these different things by using molecular methods. And I'm going to talk about another one next week. The one I'm going to talk about next week is, imagine that if one of these cancer genes goes haywire, imagine that it makes a messenger RNA, and it's not supposed to make it, that we could make a drug that could inhibit the translation of that messenger RNA so it doesn't make the protein. And I'm going to talk about that next week, because that's becoming a very, 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 very powerful therapy. So just hold on to that. But also imagine if we could block the different sites on the cell surface that's getting signaling from the cell in an abnormal way, and we could block that signal from telling the cell what to do. And that happens to be a drug called Herceptin, which was developed by Dr. Dennis Slayman here at UCLA. And that particular drug is the most common form of treatment for breast cancer in existence today. Women who take chemotherapy and Herceptin have a twofold greater survival rate than individuals that don't take Herceptin. And the way Herceptin works is it's a vaccine. Herceptin is a vaccine to one of the proteins on the outside of the breast cancer cell. And that vaccine binds to that particular protein and prevents an abnormal signal from telling the breast cells to divide. And so it's a vaccine against the cancer cell, against a specific cancer cell, in this case, breast cancer. And that vaccine, conceptually, is one protein. It's an antibody. That's all it is. It has a light chain, a heavy chain, a variable chain, constant region. It's an antibody, and was discovered because of genetic engineering. In other words, isolating these particular proteins and these particular genes, but was also discovered because of the fact that one can know what the cause of that particular cancer is. And then it was an antibody that was genetically engineered conceptually to be able to be used as a drug to combat cancer. And in a sense, and I'm not going to get into it only conceptually, it's made in the following ways. It's made using a variety of different procedures that I'm not going to really get into, but the point here is that it can recognize the tumor cell, and when it recognizes the tumor cell, it induces these other parts of your immune system called the T cell, which has this specific antibody to then destroy the cancer cell in a very specific way. This is becoming, I think, one of the most promising and absolutely most effective way to treat cancer, along with what I'll talk about a little bit more next week. Brian, you have any questions? This is really cutting-edge stuff. The landscape is really changing for many of these really serious diseases, including cancer, because of our ability to identify the genes, know what they do, identify the proteins, understand the process, and be able to genetically engineer things in order to combat them. And so this is a very, very, very powerful kind of technology. How many of you have heard of MRSA, M-R-S-A? Tell me what it is, Max.
It's a type of,
Use the microphone.
it's a type of bacteria
Speak up.
that's resistant to a lot of different types of antibiotics?
Yeah, namely methicillin, which is the only antibiotic which has been used effectively to counteract this bacteria called Staphylococcus aureus, which is just a normal skin bacteria. But if you get it, it can be lethal, and it's devastating. And so the point here is that one then has another use of genetic engineering, again of bacteria and a variety of other things, and that is either discovering creatures that make new antibiotics and or engineering bacteria to make better antibiotics. And the reason for this is that antibiotic resistance, as we touched on in this class a bit, is becoming really serious. So notice that we have penicillin, Staphylococcus aureus, almost 100% resistant to penicillin. We have it only about 32%, but going up, resistant to methicillin, which is a type of penicillin. Ampicillin, penicillin, methicillin. It's a type of penicillin. It works to kill bacteria cells by preventing the cell wall from replicating, from dividing. And so the bacteria just bursts. It's like a balloon that bursts. The point here is this resistance is very, very, very serious. So we're always trying to outwit these pathogens, mainly bacteria, with these antibiotics. And imagine living in an antibiotic-free world. We'd be in pretty bad shape, as I showed you from that curve a few moments ago. Now, I've showed you this before. It's taken from your book, and we talked about this, that antibiotics have different mechanisms of action in bacterial cells. Antibiotics don't affect human cells, they affect bacterial cells, and they're generally made by other bacteria or by fungi, and again in sort of a microbial warfare. And these antibiotics can prevent the cell wall from synthesizing, can prevent DNA replication, can prevent transcription, can involve translation, variety of different things. And we discussed this a few weeks ago. And furthermore, when we have a methicillin, as I said, it prevents the division of prevents the formation of the cell wall. So you don't have a cell wall in the bacteria. All you have is a membrane, and it bursts just due to pressure from inside the cell. The bacteria become resistant because the resistance genes are on plasmids, and the plasmids are transmitted in the population. You get resistance, as we discussed. In the case of methicillin, there happens to be a gene in the Staphylococcus genome in a plasmid. That gene actually detoxifies the antibiotic. So the methicillin doesn't work because the gene on a plasmid in this Staphylococcus bacteria encodes something that destroys the antibiotic. And so the question is, how do you come up with new antibiotics? Because you need them. Well, one of the ways in which you can do this is two ways. The first is discover new bacteria or fungi that make antibiotics. Antibiotics are made by bacteria and or fungi. So it's the idea is to discover new creatures that then make new antibiotics, and hopefully those new antibiotics can be used against bacterial strains that are resistant to existing antibiotics. And we'll come to that in a second. The other way in which it can happen is, what are antibiotics? Antibiotics are small molecules. They're organic molecules. They're made up of carbon and hydrogen and oxygen and other things. And if you know the pathway, that is A to B to C to D to E to antibiotic, and you know the genes that are involved, and you know how they work inside the bacteria, you can start engineering these pathways by adding new genes that add new little groups to the antibiotic. Let's say, for example, that you have an antibiotic which has this kind of structure, and those are, let's say, carbons, and that's the structure of the antibiotic. And let's say you know how this is made, and you know what enzymes make it. Let's say that you want to add something onto that antibiotic, and by adding something, the antibiotic will work, but the gene that the bacteria has evolved that is resistant to the antibiotic no longer can destroy the antibiotic, because this molecule that's been added now protects the antibiotic from the antibiotic resistance gene in the bacteria. Is that clear, Eden? So if you understand the pathway that gives rise to this thing, and if you can engineer a new enzyme or a new gene into that pathway so that it adds something onto the antibiotic, you now have a new antibiotic. And that's where genetic engineering can be used in a very, very powerful way. So again, it's either discovering new creatures, or it's engineering the pathways in order to make better antibiotics. And so again, how do you know this? How do you know this pathway or that pathway? Sequencing the bacterial genome, understanding what the genes do, understanding what the genes are in this pathway and what the proteins are, and then being able to think about ways to engineer a new pathway. Or alternatively, how do you find new creatures that might make new antibiotics? Metagenomics. Many of the bacteria that make antibiotics are soil bacteria. So you just scoop up soil all over the world, and you just start sequencing everything that you find in the soil, and you start looking for new bacteria and or fungi that have the potential to make new antibiotics that you discover by sequencing your iPhone, huh? Can't be that important. In any case, the point here is that you have two alternative methods in order to make these antibiotics. Okay, is that clear? The fascinating thing about genetic engineering is that bacteria and microbes are used for a lot of different stuff. Not just drugs, not just vaccines, but they're used to make all kinds of stuff. I mean, if you really knew about all the different things that bacteria produce, you'd be amazed. How about the enzymes that go in softeners of your detergent? Those enzymes are made in bacteria. And then they're put in this, downy softener and stuff like that, and it softens your laundry. How about things that are involved in baking, let's say, bread? How about things that I'll tell you about in terms of making cheese? How about small molecules like manufacturing, acetone and butanol and biofuels, and all kinds of different stuff are made in bacteria. And genetic engineering can have a huge impact on these things, and I'll give you a couple of examples. So the first thing is cheese. How many of you eat cheese? How many of you know that cheese is a GMO, in a sense? How many of you know that? Thomas, you knew that? How did you know that? Where's your microphone?
Whoa.
Ingrid, what are you going to do with all these film clips? Great. Go ahead.
Well, the bacteria that go to ferment and create the cheese are genetically modified nowadays, aren't they?
Not quite, because cheese isn't fermented. Alcohol is fermented, but cheese isn't. So how many of you ate cheese in the last 2 days? How many of you know that this cheese that you ate was made by genetic engineering? How many of you knew that? Even the stuff that you buy at this wonderful organic food store, in which you pay $10 a pound for tomatoes, in which have signs and say, genetic engineering, the next ecological disaster of the world, of which funds in the most highest amount the anti-GMO campaigns to rid the world of plant genetic engineering, that wonderful, fancy, fabulous store in which all you see is Porsches and Mercedes-Benz and all kinds of cars in front, called the Whole Foods grocery store. How many of you knew that even in there, the cheese that you eat was made by genetic engineering? And I believe you, if you go and tell those people there, they're going to have a heart attack. Because that could be the worst thing that could ever happen to them, is to eat a genetically engineered potato chip. They may actually have a heart attack. I don't step one foot in that door. In any case, that's a story for another time, over dinner. The point here is that cheese is made from milk. And in order to make cheese from milk, you have to do something to the milk proteins to get them to coagulate. You with me, Dora?
Yeah.
You had cheese lately? They have a Whole Foods up in Davis? Oh, the answer is no. That's good. In any case, I wouldn't have thought so in an Aggie town like Davis. The point here is, just joking, the point here is that you have to add something to cheese in order to coagulate the proteins that are in the milk. There's a lot of proteins in milk because obviously it's very nutritious. And one of the things that is used is this thing called rennin or chymosin. And traditionally where it was gotten was from the stomachs of cows. Maura, how many stomachs do you think you have to get a little tiny bit of chymosin to be able to use it to make cheese? A lot or a little?
A lot. I know cows have this whole different stuff.
Pardon?
Hello. Yeah. I'd say more than one.
How many do you think?
Isn't it 4?
4 stomachs in order to get enough chymosin in order to make cheese? 4 stomachs?
Oh, a lot more than that.
Probably about 4 million.
Oh, okay. I was thinking per cow.
Each cow has 1 stomach. It's actually a ruminant. It's not quite a stomach. The point here is that before genetic engineering, they had to go to slaughterhouses, get literally hundreds of thousands of stomachs, grind them up and purify this protein, even though you can't completely get it pure, and use it to make cheese. Not a very successful process. But after cloning was invented, one could then go to the stomach of a cow, clone its messenger RNA, and clone the chymosin protein gene, or the cDNA. So you could get chymosin cDNA, and then you could make chymosin in bacteria, much like you could make insulin in bacteria. And so you get tons of chymosin. Get as much as you want. And then if you go to the FDA and you say, well, I want to use this chymosin in order to make cheese, they say, well, let's look at it. Let's look at the process. Let's look at how pure it is. Well, it is pure. There's nothing else there. It does work. Doesn't contaminate anything. You don't need to kill millions of cows in order to get their stomachs in order to do this. And this is really a good thing. And so chymosin, recombinant chymosin, is used to make 70 to 80 to 90% of the cheese that's made. And so most of the cheese is then, in a sense, made from recombinant protein, this chymosin. The question really is whether this cheese that's made from this chymosin from a cow, is that kosher cheese or not? What do you think, Max? Does anyone take a guess? What do you think? It's got a cow stuff. It's got some meat. Is it kosher or not? Well, Zach, Mike?
A friend of mine was doing rabbinical
What do you
studies.
think?
I know for a
all I wanna
fact
know
that
what
it's
you
not
think.
kosher.
Pardon?
It's not kosher.
Actually, it is.
According to him, it wasn't.
Yeah, your friend's wrong. The point here is that it is considered to be perfectly legitimate stuff. Yeah. Anyways, it was a sort of an ethical dilemma. But go ahead, Albert.
Are you sure?
Am I sure?
Because I know.
Well, I could always be wrong.
Well,
But
because I know that, like,
I
for
could
example,
always be
a lot
wrong.
of Jell-O isn't kosher because it's made from gelatin, even though gelatin's made in a very similar fashion now, right?
yeah,
With
I could be
bacteria.
wrong, but from all of my reading and all of my knowledge, for as far as I'm concerned, it's okay. You know, it's not like mixing the cow with the milk. For those of you that partake in these kinds of things. The point here is that we have one of the first genetically engineered food additives, in a sense, approved by the FDA, perfectly legitimate, used to make cheese, accepted, generally regarded as safe, meaning the same as the normal chymosin. There's no labeling involved. It's not this cheese was made from recombinant chymosin. There isn't any difference between chymosin. In fact, it's purer than the chymosin you could get out of the cow's stomach, much purer, and it's used for the production of cheese for vegetarians and vegans. Let me move on and wrap up a couple of things. So the point here is is that one can then take this chymosin and ask the question, can we improve upon it? What do you think about that, Dora?
Yes, you can improve.
Maybe you can. That would be great. Maybe it can work better. And the way in which you can do that, and I'm not going to get into this in detail, I'm just throwing this out, is again another use of genetic engineering called protein engineering. And so now you design the protein in the laboratory using genetic engineering by taking different pieces of proteins that may act like chymosin. You put the DNA for those proteins together in different combinations. You shuffle them using the polymerase chain reaction. You get all kinds of different proteins now, different cDNAs that encode different amino acid sequences, although you can do this in a much more precise way. And you try and come up with a protein that's more effective than chymosin, let's say superchymosin. And this is very powerful for engineering enzymes to make them work better, and very powerful for making other kinds of proteins. And there's a whole company called Maxigen up in the Bay Area which does this kind of stuff and pioneered this particular kind of approach. In wrapping all of this up, bacteria make small molecules called metabolites. Ethanol, biofuel, butanol, biofuel, and vitamins, variety of different things. How many of you take vitamins? Made in bacteria, isolated from bacteria. How many of you think that they can be engineered to be better? You can use genetic engineering to do that. So the point here is that bacteria synthesize, there's the metabolic pathways inside a typical cell. Look at how they're all connected, thousands of genes making thousands of proteins, all kinds of chemical reactions. Derek, you're a biochemistry major, but you're just a bag of chemicals in reality. That's what makes us work. We're not that different. We are bags of chemicals, organized bags of chemicals, organized in the sense that are working in a very, very precise way, but that's really what's going on inside us. Here's the pathways leading to specific products, ethanol, butanol, variety of things, a pathogen, whatever. But the point here is that one can intercede in this process by using genetic engineering, by using protein genetic engineering. One can make better enzymes, better processes, and better end products, such as biofuels, such as making biofuels in E. coli, such as making dyes in E. coli, dyes that are something that was originally gotten from plants. Now these pathways, one a gene from E. coli, one a gene from a different bacteria, Pseudomonas, put together in the same bacteria, E. coli, using genetic engineering. Now we're making indigo, which is a dye for genes. And so the point here is that one can use genetic engineering for those things. Pollutants in the environment. This is an extremely important. Bacteria will degrade and use as a carbon source, will degrade and will use as a carbon source a variety of things in the environment. Xylenes, naphthalines, all kinds of nasty stuff. Oil, variety of hydrocarbons. One of the things that we're going to talk about 2 weeks from now is this very famous landmark case of Chakrabarty, U.S. patent 4,259,444, in which the Supreme Court said life is patentable. And that's because Professor Chakrabarty took a variety of different plasmids containing different genes and made a new pathway in this particular bacteria, Pseudomonas, and got it to degrade oil. And he patented that particular organism, and he was allowed to do so for reasons that we're going to talk about. The point here is that you can engineer these bacterial pathways and engineer them in order to do a variety of different things. The final example that I'm going to give you is we now talked about using genetic engineering to make drugs, using genetic engineering to make vaccines, using genetic engineering to make protein products for food, using genetic engineering to alter pathways that can make vitamins and a variety of things. But how about genetic engineering bacteria to deliver drugs to you. So for example, what about engineering bacteria so that they can deliver a particular molecule that can combat disease? So in this case, one has an intestinal disease, either ulcerative colitis or Crohn's. It's due to a defect in your immune system. If one can deliver a particular protein that can counteract the effects of your immune system, it can counteract that particular disease. If one can engineer a normal intestinal bacteria with a human gene that can produce a human protein that can be released in the intestine, that can reduce the inflammation and reduce the disease, that would be very powerful. This is now in development, but it's been shown in mice to be very effective. Here is a mouse. Remember, we talked about knocking out mice genes. Remember, we talked about using mice as models for human disease. So this mouse has a gene which is knocked out called IL-10, which is involved in facilitating the immune system, and that mouse gets intestinal disease 100% of the time. Now, if you treat this mouse with this stomach intestinal bacteria that has been engineered with this IL-10, the mouse doesn't get this disease because this IL-10, which is needed very, 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 very much to counteract that disease, is now given by the normal intestinal bacteria in a very, very common way. So this could be a very powerful tool that could be used for engineering garden variety intestinal bacteria in humans for delivering things that are needed by us in order to overcome certain diseases. And then finally, how many of you ever had a cavity? You know that cavities are caused by bacteria that live in your mouth, and by fermentation, they give out chemicals that destroy the enamel in your tooth. Well, what if you could engineer bacteria which couldn't do this, and let's say you could flood, using a mouthwash or a toothbrush or a toothpaste, your mouth with, let's say, engineered bacteria that outcompete the ones that cause tooth decay, you might not be able to have, you might not get tooth decay. And so this is something that's also being developed, and that is genetically engineered bacteria, which normally would cause tooth decay, but in this particular case don't. The point here is that there's many, many uses of genetic engineering, of which I've probably scratched the surface of about 5% of all the different uses that have practical applications. We've concentrated on those that are the big ones that have affected and are in the news and affect us the most, but there's a lot of stuff going on, better vitamins, better metabolites, trying to engineer bugs for biofuels and other kinds of things which are under the radar screen and which you don't use, but on the other hand are extremely powerful, and powerful enough that the take-home message is that this technology affects your life in so many different ways that you can't even imagine, and has changed so many things that most of the things that it's changed are really below the radar screen. Dora, do you have any questions?
No.
Pretty remarkable stuff, isn't it?
Yeah.
It's not just making insulin and bacteria, it's a lot of other things. And a week from today, we'll talk about some other pretty amazing things too, as it pertains to animals and humans.